Okay, that should be right.
Well, I was going to mention,
Mhm.
in case you have some virtual, before I leave, can you let me know how many of them were virtual?
Yes.
That listened to me at least, yeah.
We'll probably do this one. Share that one. And sorry, it takes me so long to get this computer set up. I have to like reload, like
No, no, you're
upload
good. It's a lot of
everything.
things.
You're okay. Yeah, it's a pain. Okay, I get some light turned on for you too. I like that dark back here so they can see the projector.
Oh yeah, for sure. No worries. Whatever works best for you.
All right, let me drag this over. So that's recording. Now it's a matter of me finding my cursor whenever there it goes. Okay. Let's do that. Chat for now. Right. Yeah. I guess you're free to start if you want to. All right, I'll have you guys, divert your attention up front. We're going to hear a little bit about SSOC and the resources that they can provide you guys throughout, your learning process at OSU OKC.
Thank you. Alrighty, so my name is Shavana Rodriguez. I'm one of 2 academic success coaches here at OSU OKC. Now, just to kind of get a little more familiar with kind of the who's kind of gone and who hasn't gone. Has anybody ever been in the SSOC before? No. Okay, well that's good because today I'll be giving you all of the resources that we provide. That'll honestly give you so many reasons to go to the SSOC. But the SSOC stands for the Student Success and Opportunity Center. We're located on the LRC second floor. We take a pretty good chunk of that floor, essentially just providing resources, coaching, and tutoring services for our students. So our hours from Monday through Thursday, we're open from 8 a.m. to 9 p.m. On Friday, we're open to 8 a.m. to 5 p.m. And on Sunday, we're open 1 to 5 p.m. So we try to accommodate to as many schedules as possible. I do want to emphasize these are the center's hours. These are not the tutor's availability. If you want that information, you can go in the OSU OKC SSFC webpage so you can see the semester's tutors and their availability for each center. So in regards to resources, we have first of all, just the space. It essentially is really great to study for both like individual or group study environment. We have couches, whether that's like individual couches or like group couches that you can use, as well as we have one big TV in case like you want to practice a presentation or if you want to see your work a little bigger so it can kind of help you. We have plenty of space and like accommodation in that sense of making sure that we accommodate for both small, big, individual study for students to be able to use. So we also do have a reservable conference room. With that one, it's also catered to our students. And if you're involved in a student organization, you can use that to meet and book that space. As well as we have a reservable testing room, which is catered towards basically making sure that you have a place on campus where you can take a test, and whether that's if you don't have a quiet place at home where you can focus, you can definitely use that testing space, as well as if you don't have lockdown browser on your devices, that computer and testing system for that room is accommodating to lockdown browser. Another thing that I did want to include in regards to how to reserve both the conference room and the testing room is by emailing okc.tutoring@okstate.edu. If you would like, whenever I'm done or during my presentation, I have some business cards for the STEM Center and the STEM lab and the Writing Center. The email that's in those business cards is the same email. You can ask any questions or reserve those resources that we have. As well as I have myself and Michaela, who's our other academic success coach here, that you can contact us for anything you might need as well. And all of the information I'm presenting today is available on rack sheet with a QR code to our website, if that makes it easier to remember. You can come and get it while I present. We also have some goodies. We have some pens, some Post-it notes. You're free to grab, but if you don't want, it's totally okay. Okay, so I've mentioned the resources. Next, I want to talk about the tutoring centers. So I know this is a science-related course, so I definitely want to emphasize the use of the STEM lab. It's catered towards science and math-related courses, whether that's physics, biology, chemistry, or calculus, algebra, or other math. Sorry, y'all. I'm very much not a math person, so I'm like, I don't really need to remember math myself. So really all the courses, science and math related that we have at OSU OKC, we have the tutoring for it essentially. And we want to make sure that you also have like study tips in regards to how to study for certain classes. One of them being for this class, Tin is really good at science courses, who is one of our full-time tutors at the STEM lab. And Brandon, who is a part-time tutor, he's really great at math courses. In case you might need more support in math and science, they're happy to help. As well as just in general, tutoring is kind of like from an outsider perspective, if you're not in like academics, it could be like, what's the word? Like maybe like something you're shy to go to. But the students that best perform are the students that seek out these resources. So please like just come in regards to just getting that extra support, because it's better to have support than no support and you're just drowning in whatever class you're in. And that includes myself for academic success coaching, but I'll get to that. But for the STEM lab, it is walk-in based. So as I mentioned before, you can access their schedule on the website. But if you just want to go into the writing, the STEM lab, it's walk-in based, so it's kind of first come, first serve in regards to like when you get help. But essentially, that's kind of the STEM lab and what they can offer, just support and making sure that you're understanding the concepts as well as you can apply them, and just study skills that you can use for math and science courses. Now, for the writing center, they can help with all subjects as long as they're writing related assignments. So that can range from all classes, because most classes you'll need to write a paper in the future. And they'll be able to help you throughout the writing process, whether that's brainstorming up to like your final proofreading before you turn it in. We do have an essay drop-off service, which essentially, if you are unable to come into the writing center, they'll be happy to just receive that through email, and you'll get the tutor's notes about your assignment. Once again, if you get one of the business cards, you can just send it to that email and we'll make sure it gets to who I need to get to. Let me see. Then, as mentioned before, I'm an academic success coach, one of two. And essentially academic success coaching words. What's it called? Is essentially making sure that you get personalized assistance with navigating college. So some of the things that we can help with is like time management, organization, how to study, how to take notes, how to prepare for a test, how to deal with stress management, especially test anxiety is one big thing that's going to come out during midterms and finals. So it's something that's common, and we want to make sure we walk you through how to best navigate that, and just in general college, because it is different no matter if you haven't been in college for years or you're just coming right out of high school. Like college, no matter what background you come from, is an adjustment, and we are making sure that we can help you with those adjustments. So how you can visit either me or Michaela, you can walk in. If you see that door and I don't have an appointment, you can easily just walk in. Of course, if you want like a catered time where no one's gonna interrupt us, I'd really recommend reserving an appointment, which once again, you can do that through the okc.tutoring at OK State. But I also brought my business card with my personal email in case you need anything. You can reach out to me, and it has my phone number as well. But what I was going to mention is we have 30-minute and hour sessions. So we accommodate to all schedules in that sense. If you are tighter on time or you want to really talk about it, we have that hour option. And we do both in person and online. So just any way you can reach us, we will find a way to reach out. So yes, that's a little bit about really everything that the SSOC has to offer. I hope after this presentation, you kind of get that encouragement to go and get support, even if it's like the most minimal amount, it's better than where you first started. So yeah, does anybody have any questions? Okay, already, like I said, my business cards are here, as well as the Writing Center, the STEM Lab, and Michaela. The information I just presented is right here in rack sheet form. If that's something you want, please go ahead and do that now.
Yeah, if you guys want to come up and get any of the resources now, she's gonna take them with her when she leaves.
Yes.
So they're not gonna be here. It's no big deal. I would say if you guys found that when you took the test for the exam, the first exam for this class, and you were kind of shocked at your ability to apply that information that you studied to the exam because the questions were different than what you were expecting, the wording was different, test anxiety got to you, you thought you studied enough, but then you realized that you didn't, whatever it is that was your problem, they can help you guys with that stuff if you don't know where to start. And it's free. So there's no, yes. It's completely free. There is absolutely no reason why you shouldn't take advantage of what they have to offer. At least go check it out and see what they can do for you. There's no reason not to.
Most definitely. All of our services are free. And yeah, once again, just awesome.
Yeah, I mean, I really honestly, it's one of those things I don't know.
Yeah,
why people
whatever's
don't take
there, you
advantage.
can take. Oh, that's a script for the rack.
Yeah, I noticed that. I'm sorry.
It's okay. I was like, I don't think you want that. All right, perfect.
Yep, you do what you need to do. Thanks for coming by. Yeah. All right, guys, go ahead and get, oh, there's nobody joining us online, by the way.
Okay.
It's just these people. 6, 7, 8, 9, 10. I have 16. Does that seem right?
Thank y'all so much for y'all's time. Thank you so much once again.
Yep, thanks. So while you guys are getting signed into the Kahoot, does anybody have any questions about any of the material or anything? Yes. I mean, I'm better than I was yesterday, but I'm not good. Like, and like the more that I'm, you know, standing up here doing stuff, the less good I feel. But you know, I'm not going to stay there. Even if I sit there, I'm going to get up and start walking around. It's not a big deal. And this is honestly, this is my favorite chapter. So I really just didn't want to miss it either. I did good on Monday, or not Monday, yesterday, Wednesday, with the recording for this. So I could have had you guys watch it, but it's just one of those things I like to, it's fun. That part's fun for me. If I'm going to be sick, at least I get to do a fun thing. Well, yeah, I'm just really drained and I can like feel the fever coming out of me. So, but I can tell the antibiotics are working. It's just not as fast as I thought they would be.
Is it pretty painful?
Yeah, like my back, it's like I can feel it in my kidneys. They're uncomfortable all the time. And like when my dogs, because I have little dogs, so they'll like, you know, jump on me and want to lay with mommy and whatever, but like they'll like be on my back and I can feel it, like the pain in my kidneys, just like the inflammation of it. It's, I don't know, I've never had a kidney infection before, so it's like been a learning experience.
I just had
Yeah. Yeah, I've only ever had like typical UTIs, you know, bladder infections and stuff. So this is my first time that's progressed that far. The doctor, like literally, I didn't have any of my typical symptoms for UTIs. I didn't have any blood in my urine. I had a little bit discomfort urinating, but it wasn't like, you know, really cloudy or anything like that. maybe an indication that could have been something, but not like kidney infection level. And so I went to the doctor and he's like, yeah, I'm pretty sure like the reason that this and that and whatever, you have the fever, I'm pretty sure you have a kidney infection. And I'm like, what? Okay. And he's like, yeah, why did you let it go this long? And I'm like, what are you talking about? Like, I didn't know that was what was happening. So it's probably just a different kind of, bacteria than what I usually have or something. So it is what it is. What can you do? I mean, just taking the medication, that's all I can do. So let's go ahead and get started with this. If you guys haven't gone through chapter 9 or 25 yet, then this is over chapter 9, which is what most of 9 and 25 are over. So good luck if you haven't gone over it yet, but that's what we're going to be playing through. And if you haven't looked through chapters 9 and 25, the video that I had posted there, it only covers the stuff that you will be tested on the exam. So I would, and it takes out stuff that's on the study sheet that you might not need. All right, so CHOMPS is used to refer to elements needed for life. What is the S? All right, so that is going to be sulfur. This is just the 6 most common elements, like at the top of the list, as far as it'll be carbon, hydrogen, oxygen, nitrogen, phosphate, and sulfur. And it is literally the symbols for the periodic table of elements. Selenium, it's another element that's on the table. It technically falls into the metal category, but it's just like one of those supplemental metals that you just need a little bit of in your diet. Yeah. You can buy, like, there's crystals you can get that are like selenium crystals. They're like selenium oxide or something that people use for, I don't know, crystal people use them. I don't know what for. I think it's for like purifying or something. Which of the following is heterotrophic? Hetero, of course, that's going to be different stuff. Troph, if you guys don't remember, that's like trough, like eating. We're doing 2 labs today, y'all. Yes, we do not skip labs in science. So when you miss a lab, you got to double up somewhere, unfortunately. We'll be kicking out part of the lab. We will not be doing the pond water, probably, unless you guys just have time or want to, or have time on a different lab and want to. I don't know, but it won't be required part, is what I'm saying. The pond water part, which is the fun part, unfortunately. But all right, so heterotrophic. The heterotroph versus the autotroph, and then we have phototroph versus chemotroph. These are things that I would highly recommend going through the lecture over, but it has to do with where you're getting your carbon or where you're getting your energy. And so this is a carbon-based concept. So are you getting your carbon from eating other organisms that already have organic molecules, different foods that are involved in that, right? Or are you an autotroph that creates your own organic carbon compounds from inorganic sources, which is trees and crap like that, right? If you're photosynthetic, then you're using inorganic carbon dioxide. The mouse isn't. So that's what that's getting at. I know a lot of you haven't gotten to that chapter yet. It's okay. It'll make more sense when you see it. All right, saprobes. What do they do? This is actually a term we've talked about before, but it came back up in this chapter. All right, so these are our decomposers. Again, just another term that we need to refamiliarize ourselves with. Diffusion of water molecules through a cell membrane down, or what we consider with the gradient, not against it. What will we call that? So diffusion of water. All right, so this is called osmosis. Osmosis is the term for diffusion of water itself. This is important in cell membranes because our selectively permeable, lipid bilayer membranes, they can only allow water and other very small nonpolar molecules to cross across naturally. So when you have to deal with things like high salt outside of the cell, or very, very like low salt compared to the inside of the cell, you have to be able to get water across to accommodate the difference there. Because the other molecules, the salt can't cross, right? That's the point. What is the solute in salt water? Like this one should get you down to at least 2 choices, and then it's a matter of knowing the term. And yes, the solute is salt and the solvent is water. That came up again in this again. I know it was in the chemistry one, but hey, it's nice to have doubling up, I think. Okay, solute concentration outside the cell is the same as inside. What is the term for that? It's equal inside and out. All right, so that's isotonic. I'm glad you guys are familiar with that term. Equal on the outside and on the inside. And then obviously the other 2 choices would be hypo or hypertonic. Hypotonic, the tonic outside of your cell, right? That's how I always think of it, right? Outside of it is going to be low versus high for hyper. Okay, so that's what they're talking about there. Is outside of the cell, what's going on? Right, facilitated diffusion. What do we need for that? All right, so it's going to be carrier proteins. It's exactly right. Facilitated diffusion, we're still having diffusion going on. So it's going to be a natural movement with a gradient. Diffusion being something like you have a whole, I always say like a big old net full of bouncy balls on one side of an area. And then when you open the net, it wants to equal out, right? It'll spill out and equal out all over the place. That's diffusion, just like if you had a dam that's holding back a lot of water, the water wants to get out of that dam, right? So if you let it open up, it's going to go to equal out on both sides, right? So that's the idea of diffusion in general. When we're talking about osmosis, we're talking about water being the thing that helps even things out. And then when we're talking about facilitated diffusion, that is like when you have a dam and you put a hole in the bottom and it can only go through that hole. We're letting the water through. It can't go through the cement, right? We're giving it a place to go through, but it's still moving by diffusion. We're not forcing it against its will, right? So that's facilitated diffusion. You don't need energy, you just need a channel. So now we can move things through the membrane that normally wouldn't be able to. That's what those are for. All right, next. For active transport, another form of how we get molecules across the membrane. What is that? I got it, man. All righty. So we're going to be moving things against the gradient. So that's like putting water back into the dam. How are you going to do that? You're going to have to put energy in to fight that force that wants to come out, right? So that's going to require energy. That makes sense. We're moving against the gradient. Yes. And it's usually done by transport proteins. Yes. There has to be some way to get through that, right? The thing that's controlling it. So that's why the answer would be all of them. Active. You can think of any time that you're being active, you need to use energy to do that, right? So that way. All right, the type of symbiosis where one organism benefits and the other is harmed. I forgot I even talked about this in chapter 9. I don't know why you get 1,000,000 seconds for this one, but you do. Sometimes I don't pay attention to that. I don't want to wait the whole other next time, so I'm going to go now. Okay. So it's parasitism. Most of you guys got this, right? So that parasite is benefiting from feeding off of its host, and that host is being harmed because of that. That's not just going to be ticks or fleas or lice or whatever other nasty thing you want to think of. That would include any of our pathogens as well, because they're all taking advantage of our cells and they're benefiting by harming us, basically. So viruses, bacteria also fall into that category when they're doing that. All right, last question. The lag phase. We're growing bacteria on the growth curve. What is the lag phase? All right, so this one again is like just being, familiar with the terms. The lag phase is where you just introduced your bacteria into your culture. Is it growing a whole ton at that point? No, right? It's just acclimating to its new environment, maybe copying new DNA because it's like, oh, we have nutrients. First, it has to copy DNA and make structures, right? And like make what it needs for new cells. That period of pause before we start really dividing and making new cells and shooting off, that's the lag phase. The next phase is called the log phase, like logarithmic or exponential, same thing, okay? That's where we're really growing, exponential growth. Then we have the stationary phase, where that's going to be growth has stagnated. Maybe growth and death is equal, or we're just not growing as many cells because there's not as many resources, things like that. And then we have, of course, the death phase, where we're going to be dying off because toxins are being produced because of waste products that have been made from dying cells and whatever. So those are the 4 phases of the bacterial growth curve. That's that for this Kahoot chapter. So we're going to kick it into chapter 10. Let this sit briefly, but then we got to get going with chapter 10. We're probably, and I got the sticks out, but y'all, we're probably not going to have time again just because of we had to have the talk in the beginning. All right, so good job, good job. All right, bring this up. All right, so chapter 10. Chapter 10 deals with microbial metabolism. Really, honestly, we're going to be talking about metabolism, basically how you or any other organism, generally speaking, makes energy. So metabolism refers to all of the chemical reactions and physical workings of the cell, and essentially how it's going to be making things, breaking things down, getting energy out of it, anything chemically that's changing in the cell. That also does translate to what's going on in your body overall, based on what systems need what energy and what needs building and everything like that. So yes, this does talk about the macro as well as the micro, but we're going to be focusing on the micro of all of this. 2 different things that will be going on in metabolism are anabolism and catabolism. So these are breakdowns within the metabolism category. I always start with catabolism when I'm describing it because I think of cats. So that's the way that I remember this. If you have a cat or you know about cats, y'all, then you know if you have like a sofa or a chair that you love, they'll destroy it. They'll claw at it and rip it apart, generally speaking, right? So I think of cats being destroyers, so they're breaking things apart. Catabolism, where we're breaking the bonds of our larger molecules to release energy. Anytime you're breaking a bond in chemistry, you're releasing that energy that was contained within that bond. Maintaining a bond, it's holding energy. When we're looking at anabolism, this is biosynthesis. So making new bonds, making new molecules. So you're going to have to put in energy to build. So in anabolism, because it's not catabolism, we know we're building, then it's like building even like a building or something like that. So if you're building a new tower out, a new Devon Tower 2 or whatever out there, you're going to be putting in a lot of energy and a lot of work into building, right? Whereas if you have a Jenga tower and you just kick it, like it probably doesn't take any energy at all to break it apart, right? So that's the idea here. Anabolism, building things takes a lot of energy. Also think of anabolism, if you've ever heard of anabolic steroids, that's exactly what that is referring to. Anabolic steroids are steroids that you would take to build muscle, to help promote building of muscle. And that's why they're called anabolic, because they're building, okay? All right, moving on. So the whole idea here is going to be use or making of ATP, typically. That's what we're referring to here. So anabolism, we're going to be using the ATP to drive our building processes. Catabolism, we're going to be hopefully making ATP, or at least getting molecules made that can eventually lead to making ATP. We can also release energy in the form of heat. So if we're breaking bonds, sometimes we'll just lose it in the form of heat, which that's energy as well. Anything that helps speed up a chemical reaction is known as a catalyst. Catalysts in any chemical reaction help overcome activation energy. So that's the energy required to get that reaction going, right? To overcome that hump that we need to put in that much energy just to make, A go to B, So that's a catalyst. So for our purposes and what we're learning about here in metabolism and microbiology, we're focusing mostly on enzymes. Okay, so it's the same concept, but enzymes are organic catalysts. So they are things that are used by the cell to make these things, go, but they are still catalysts. Okay, there's other ways we can help with, promoting these chemical reactions to do what they're supposed to do to help overcome that activation energy. If we're not using enzymes, we can increase heat. So introducing heat can cause, interactions to speed up. We can have increase of reactants to help force it towards that product. And then we have adding a catalyst. Well, it says adding catalyst, but it could be other catalysts besides just enzymes. Depending on what chemical reaction that you're talking about, you might go with one or the other of these. But we're going to be talking about enzymes today. So enzymes are typically going to be proteins, typically, okay? For all of our purposes, anytime I'm talking about an enzyme, they're proteins. They usually end in ase, right? And they bind on something called a substrate. The thing that they're going to act on is called the substrate. All right, so when we're talking about DNA polymerase, ACE, that's an enzyme, right? So it's a protein that's going to be polymerizing DNA. So typically what you could think of is it's using the template strand and adding on complementary nucleotides as it goes along. So both of those could be considered substrates, or you could just look directly at how it's handling the nucleotide and whatever. So that's a little bit more of a complicated one, but that's what we're talking about. That's the things that it's acting upon. All right. So these enzymes are not used up by the reaction. They can be changed within the reaction, but they can usually be changed back and be reused over and over again. So we don't have any loss of them. We have two different kinds of enzymes. These protein molecules can be either simple enzymes or conjugated enzymes. If they're simple, they're just proteins. But if they're conjugated, it means they're protein with something that helps it function, a non-protein molecule. So here are our conjugated enzymes. When they're whole functioning enzymes, we call them holoenzymes. I think that's nice, even though whole is spelled with a W, still sounds the same. There you go. That's your word for the whole functioning enzyme, all right? The holoenzymes. The holoenzymes consist of apoenzyme and cofactor of some kind. The apoenzyme is the protein portion of the conjugated enzyme. The non-protein portion, the part that is helping the protein portion function, is called the cofactor. And that cofactor can be organic or not, inorganic, okay? So if it's organic, it has its own term. We call it the coenzyme. And then if it's inorganic, it's usually going to be metal ions. Think of hemoglobin. So hemoglobin is a protein, yeah, and it has a molecule in that is metal, iron, right? It's like a ferrous substrate thing. I don't know what exactly it is, but I know it has iron in it, y'all, and we all know that. So that iron in there would be like its cofactor, its inorganic cofactor, all right? So you would need to have that. That's not the thing that the enzyme is acting upon. That's the thing that that enzyme would be actually needing in order to function. If you don't have iron in your hemoglobin, it's not going to carry oxygen, right? So that's the idea there. You have to have that present in order for it to function. That's a pretty decent example of what's going on, comparatively speaking anyways. So yes. So our organic molecules that can be coenzymes, so it's easy enough to think about the metal guys. Our organic molecules that can help out with this are coenzymes. Often those are going to be vitamins. So vitamin B, vitamin A, vitamin D. That's why we need these in our diet, is because they help a lot of our enzymes function. All right, so when we look at the apoenzyme portion itself, the catalytic or active site, I'll usually only call it the active site. The active site is where the substrate that we're acting upon actually binds at the protein, okay? And each enzyme has a different, and if you're comparing DNA polymerase to RNA polymerase or whatever ACE you want to, they have a different primary structure. So that's a different amino acid sequence from one another. They have different variations in folding that can make them different. And they have a different active sites that can help their function be different as well. So there's a lot going on with these guys. And I'm going to tell you right now, far more important in enzymes and in these protein structures that are performing these functions, far more important in these structures is the actual structure as opposed to the chemical makeup. I know that we've learned all in chemistry. Chemistry has all these exact, chemical things. They're going, this chemical with this chemical goes to that chemical, or this chemical can be broken down into these chemicals. And it's this exact makeup of chemicals. That is not how biology works. Okay, it's based on generalizations a lot of the time. So if you have a protein that relies on this amino acid sequence, that could be very important. Or maybe in some parts of that protein, as long as you have a charged molecule there, you're probably fine. You're going to fold and operate correctly. You're never going to know until you work with it, right? But that's the idea of how this is going to be. So the structure is incredibly important in these guys. And I'm going to show you why in a little while when we talk about how these are inhibited. But you can see here, they often work like a lock and key mechanism. They have an exact shape that's been folded into them by their structure, whatever's dictated by those hydrogen bonds and folding and everything like that, that only certain things can fit in there. And like I said, it's not just going to be chemical interaction, it's also structural. All right, moving on to our cofactors. We've already talked about our metallic cofactors. There's a bunch of different ones you can have besides just iron. And I'm not going to focus on this a whole lot other than we already talked about this and said kind of what they can do. But they can participate directly in the interactions because they aren't the enzyme themselves. They can be used up, but the enzymes won't be. All right, then the coenzymes, like I said, these are organic compounds. These are typically going to be vitamins a lot of the time. It can be other molecules as well. And they're just going to assist the protein portion in doing its work. They can also carry parts of molecules from one part to another part or whatever. And I'm not going to necessarily test you on that. I would just be aware of cofactors can be inorganic cofactors, and they're just called cofactors, or cofactors can be organic versions, which we would call coenzymes. But if I were to ask you, are coenzymes cofactors? The answer is yes. So that's a broad umbrella term for anything that helps the enzyme. Okay, so don't get caught up in that. The 6 classes of enzymes. All right, so we have 6 basic classes of enzymes that your enzymes can fall into. And if this isn't going to hint at what's coming up in this chapter, I don't know what would. Oxidoreductases. Y'all remember redox reactions? Please tell me you remember what a redox reaction is, because we're going to talk about it. Oxidoreductases are enzymes that are going to be participating in redox reactions. Okay, and basics before we get into it. For a redox reaction, these are reduction and oxidation reactions, by the way, it's abbreviation. Reduction and oxidation reactions are just talking about exchange of electrons. That's all. We'll talk about what are, what's reduced and what's oxidized in a minute. Okay. Transferases are enzymes that will just transfer a group from one place to another, one substrate to another substrate. Moving a phosphate from blah, blah, blah phosphate onto ADP to make it ATP, for example. Okay. Just moving it. Hydrolases, they're going to break bonds. And because you broke the bond, we're also going to cleave water to assist with that. When you're breaking off different sugar molecules from starch, starch is a repeat of sugar molecules. It's a polysaccharide that's made to store sugars. If you're breaking off those sugars off of there and breaking it down, that's done by an enzyme called amylase. And amylase is a hydrolase because when you break off that sugar, you don't just leave the empty molecule, right? You have to have introduced some other bond to compensate for what you've taken off. And that's what we do with water. We'll split it. One side gets an H, the other side gets an OH, typically. Okay. That's how hydrolases work. The opposite of hydrolases basically is the ligases across the way. Remember we called about DNA ligase. Remember that one. So that falls in this category. Obviously, we're catalyzing formation of bonds. Now we're building. Yes. So if we're building, we're going to have to add energy. That's ATP. So that's why that's there. But we're also going to remove water. So this is literally the opposite of the hydrolase. So if you're building, you're creating bonds, you can usually remove an H and an OH and make them a water. And that's typically what's going to be going on with a ligase. That's what DNA ligase does when it joins the nucleotides together. All right, above those guys, we have the liases. Those are for double bond guys. I'm never going to ask you about them. And then we have the isomerases. These just change substrates to different isometric forms. And when we're talking about isomers, if you've ever heard of like, I don't know, D-lactose or L-lactose or D-alanine and L-alanine and that sort of stuff, those are just orientations of those molecules. And they're typically going to be, relatively speaking, mirror images of one another. The isomerases can change them from one to another. So that's what those guys do. And obviously the most important ones here are going to be our oxidoreductases, transferases, and then our hydrolases versus ligases. Yes. Yes. So the oxidoreductases, those are enzymes that are involved in what we call redox reactions. So reduction and oxidation reactions. And that's just the transfer of electrons. And it says here, dehydrogenases transfer hydrogens from one compound to another. And we're going to get into what that means and how that plays into electron exchange. But we've been referring to hydrogens basically as H pluses a lot of the course. But when you have a hydrogen that's bound onto something, like let's say methane, CH4, those H's are attached with electrons. And so when we're talking about transfer of electrons in biology, they're not usually free electrons floating around. They will be at some point when we get to it. But typically you're moving them around in association with a hydrogen. Because when it's not H plus floating out on its own, it's going to be bound to something and has to be associated with its own electron, like it typically would be if we were looking at it on the periodic table of elements. We know H is a single proton, that's why it's one, and it has to come with one electron. Otherwise it's H plus. That's not the normal hydrogen. That's what we call basically a proton, essentially, effectively. So we'll get into that a little bit, but we use the H, the hydrogen, as a means of transferring electrons as we're going through our chemical reactions. That's what this whole chapter is going to be about. So I hope that you guys can follow along with that because it gets, I don't know what else to say about it, hydrogeny, okay? So oxidoreductases, this is our next slide. It does basically break it down a little bit. Oxidation versus reduction, all right? So oxidation is loss of electrons, reduction is gain of electrons. If you're just gonna write that down and hope that that's gonna get you through knowing which one is which, I guarantee you will not remember. Guarantee you, you will not remember. So here's how, huh? Right, exactly. So it just depends on how you look at it, but because I could explain it, if I explain the whole behind it and like things that are reducing agents versus oxidizing reagents, the words don't apply the same as like what has been oxidized and what has been reduced. That is usually what people think of with oxidation reduction, but that makes it make more sense if you can understand it from that side. Other than that, without getting into that, because that will just confuse people if I start into that. So we're just going to stick with having to know what oxidation and reduction are. Okay. So here's how I remember it. Okay. When I took AP Chem, there's two ways you can remember it. This is how I learned it. Leo says grr. Leo the lion, right? Says grr. Or you can remember as oil rig. And I'm going to go over both. Oil rig or Leo says grr. So L is loss of electrons, which is put E minus for that, is oxidation. G is gain. And then electrons is reduction. If you have lost electrons, you're oxidized. If you have gained electrons, you're reduced. That's what that's telling you. And you don't have to worry about the concept behind it. Remember that, and it already tells you the answer. You don't have to worry about why or how or if or what. If you lost electrons, you are oxidized. Okay, and when we're talking about oxidation and reduction reactions, you have to have both. Always, 100% of the time. You're not losing electrons into the ether somewhere. They have to be accounted for. So what is donating the electrons and what is gaining the electrons? If you're having that reaction go, you have to have both. So which one is which? Always. So the enzymes that are helping this are called oxidoreductases. Like that at least is understandable once you get to this part. Anyways, oil rig, okay? Oxidation is loss. And then, of course, reduction, please, is gain. Whichever one you like better. Okay, I use Leo says GER because that's what I learned in AP Chem. Okay. So that's basically the gist of oxidation and reduction. And what you need to know about it in here, we aren't going to go through chemical reactions and me tell you, do this chemical reaction and show me which one's oxidized and reduced. I hope that you already did that in chemistry. And I'm way more concerned about the concept than I am about you following each electron at every single little tiny step and telling me whatever. The concepts are more important to me. When we're going to get into this stuff, these molecules down here, NAD and FAD, don't worry about what they stand for. Okay, these molecules, because we're going to be seeing them a whole ton in a minute. These are coenzymes. So that means that an enzyme relies on them, and it means that they're organic, right? They are carriers of what? Of electrons. So they're going to be carrying electrons for us. And I already told you we're going to be looking out for carrying electrons in the form of hydrogens. So these guys typically are going to look like NADH whenever they are, if they gain an electron, what would we call that? Reduced, right? So if they're reduced, NADH, and FAD usually does FADH2. So these are their reduced forms, and these are their oxidized forms. And typically also when we have NADH, it also comes along with an H plus just because of the chemical reactions that are going along, but not always, but just sometimes. And those are important too, but we'll come to those in a minute. So if it has an H associated with it, that is an electron. That's how they're carrying that electron. Cool. Okay, so let's talk about different kinds of enzymes. Exoenzymes versus endoenzymes. I hope that they speak for themselves to you guys, but the exo, outside enzymes, these are enzymes you put outside of the cell to break down things outside of the cell. Like we were talking about saprobes before. If you can exude enzymes out of your little cell body, and then, after those enzymes have broken down the food, then you can absorb up those littler nutrients. So that might be useful. Also, if you're making certain toxins that are enzymes, which I think we're going to talk about in a minute, and then endoenzymes are enzymes that are inside of the cell, doing work inside of the cell. Any of them, DNA polymerase, RNA polymerase, all of these guys fall into that. And most metabolic pathways, including all the ones we're going to talk about today, fall into that category. We either have constitutive enzymes. Constitutive means that it is part of it always. Okay, like constitution, like that's just part of it always. So they're always on. They're always just the amount that they are. They're not going to be increased or decreased. They're just always there in that amount. And then we have regulated enzymes. These are the ones that can be turned on or off or increased or decreased somehow in response to the need by the cell. We learned a little bit about the concept behind this when we learned about inducible operons and repressible operons, right? But any protein can be controlled that way. It's just in our cells, if you're inducing or repressing, it's not an operon, it's just one gene. That's the difference, okay? So an operon is just a set of genes all controlled at once. That's the only difference. Bacteria do that, we do not do that, right? We have to do it one by one. But we can still regulate our enzymes a similar way. So obviously if you're constitutive, no matter how much of the red substrate you have, the purple guy is going to be in the same amount, doesn't change. For our regulated enzymes over here, if you add more yellow substrate, we're going to increase the amount of the purple guys. Or if you have a ton of the purple guys, but you're not having as much yellow and it's being used up, we're not going to make as much. Exactly like we were talking about with the operons. It's just you can look at it as one gene this time, okay? Some microbes use these enzymes like the exoenzymes, like I was saying, for their benefit, not just for living in general, but also as pathogens. So we would consider that to be a virulence factor. Anything that helps an organism to cause disease or establish infection, we call that a virulence factor. So that's a virulence factor, the thing that does that. Pathogenicity refers to the ability of that organism to cause disease. Okay, so not the thing that does it, but the ability of that organism to do it. So these virulence factors, because they contribute to pathogenicity, causing disease, right? A lot of them are toxins, and some of them, these toxins are enzymes. And like it gives examples down here, streptokinase, streptolysin, elastase, collagenase. I don't know what all of them do exactly like at a chemical level, but I'll give you an example of 2 of them. Penicillinase, ACE, enzyme. So we know it's an enzyme. What do you think it does to poor little penicillin? It degrades penicillin. This is an antibiotic resistance gene. If you have the gene to code for penicillinase, then you can break down penicillin, and that's antibiotic resistance. That's how antibiotic resistance works. It's not like you just carry a gene that's labeled antibiotic resistance and then you just are resistant, right? You have to do something to be resistant. So this is one of the things you can do. So another gene that some organisms have, this is part of some organisms just naturally, it's not necessarily an extra thing. Collagenase. So again, we have an enzyme, and what do you think it does to poor little collagen? It breaks it down. So it's going to break down collagen. So what does that do for the organism? Well, a lot of your extracellular matrix and what holds your cells together and your organs and everything like that, a good portion of that is going to be things like hyaluronic acid and collagen and other molecules like that. That's what they do normally. So they maintain the structure of your tissues. And so some organisms like causes strep throat can break down those things like collagen or hyaluronic acid, and that helps them get deeper into your tissues. So it's for invasion, right? So it's an invasion tactic. Cool stuff, right? We're going to learn all about that stuff and which organisms do what in Unit 4, because we're going to be going over all of our organisms that cause each disease and applying all of this knowledge that we're learning and determining why each one causes the disease that they do. We're not just going to be like, hey, this causes a hemorrhagic fever. Isn't that cool? Move on. No, y'all going to learn about how. So that's what Unit 4 is for. So when we get to it, it's fun still. It's a little bit more relaxed. It's more like a memorization chapter at least. So if you're good at that stuff, it should help. But or unit, I should say. Anyway, so sensitivity of enzymes. We learned all about how you can break down DNA hydrogen bonds with heat. We did it over and over again with our PCR, right? You can do the same thing to proteins. They depend heavily on their hydrogen bonds to maintain their structure. So of course, if you introduce heat, you can break them apart that way. Similarly, when you introduce a bunch of H pluses, it interferes with how those hydrogen bonds are going with each other. Instead of going with each other, they might be with the H pluses, or if there's a bunch of OH negatives, so taking away the H pluses, that can affect how your H pluses would be binding across the way as well. So all of those things, temperature or pH or other chemical interactions can denature the protein. So that's dissolving its functional state, its natural state. And now it can't function the way it's supposed to. Some proteins, if they've been denatured, can go back to their functional form, but that's unusual. When you think about cooking an egg, if you've cooked your egg on your cooktop, you've denatured the proteins that are in the egg. That's what you've done. That's why it changes the way that it does. Can you go back to the original? No, right? Have you ever tried? I mean, probably not, but you can't, right? It's done. Like that's it for the egg. So a lot of times proteins can't go back. So that's what's going on with your egg. In case you ever wondered, you're denaturing its proteins. So let's talk about how our enzymes can work in systems. So we don't just have usually one enzyme. It's like one enzyme does this, and then a next enzyme does the next step, and so on and so forth. They can be linear. A enzyme here makes B happen. Enzyme here makes C, and these might be different enzymes. Enzyme 1, enzyme 2, and whatever. Cool, that's easy enough to imagine. We also have cyclic enzyme systems where you might have U going into this reaction combines with V, and then an enzyme acts on U plus V to make W or whatever. You like can put in energy or take out things along the way, and then you remake V, and then the next U can come along. Okay. If you think we're not going to talk about that, y'all, we're getting into Krebs cycle. If you ain't heard of a Krebs cycle, you're about to. Okay. Because that's what the Krebs cycle is. Anyways, and then we of course have branch, and these ones are obvious if you really look at them for what they are. That is just taking something and maybe breaking it into 2 parts. So we turned it into this, now it's breaking into 2 parts. This goes into this part and this part, and they go down their own path. Wow, that's not that impressive, really. So, or you could take these things, we made C, and we made from X, we went to Z, put them together, and that makes M. So these are not crazy concepts here on those ones. The cyclic is sometimes difficult for people. All right, how are we going to control how these enzymes function? We've kind of already talked a little bit about the genetic end of it, and we're going to retouch on that in this as well. But let's talk about how you actually control the enzyme itself and how it's actually functioning in practice. Okay, so this will be competitive versus non-competitive inhibition. In competitive inhibition, which is what we're starting with here, we have something binding at that active site. So if you are an enzyme that binds onto a nucleotide, right? And normally you bind on, let's even do something you can actually see. You bind onto a nucleotide, okay? And you're holding onto a nucleotide, and that's your normal thing, what you will normally act on to do whatever you're doing, okay? If something else comes in that mimics it, that isn't a nucleotide, but binds in that same spot, in that active site for that substrate, but now we can't fit our normal thing in here. We're blocked in that spot, specifically literally in that same spot. That active site is blocked by the binding of this thing. So this is the inhibitor in that sense, okay? So our inhibitor binds there instead, and that effectively turns off the function of that enzyme while it's bound. And it can be devastating for the cell if you cannot use that enzyme, right? So that's how that would work. That's called competitive because we're competing for that active site. Okay. Next one is non-competitive inhibition. Now we're talking about something binding at a regulatory site. So if I'm walking along and I'm a protein and I have to be folded this way to interact with my nucleotide, right? And I'm holding onto my nucleotide and we're happy and we can do our thing normally. Let's imagine that something else comes along. The regulator comes along and binds at its regulatory site. It ain't here. It's not at the active site, okay? This time it binds somewhere else. Let's say on my back, right? And now because it's bound on my back, pretend that it is bound, okay? Because I can't hold it there and do it at the same time. Now maybe I go like this and I can't hold it causes the protein to fold differently because of chemical reactions and all that stuff. So now we're folded differently and now we can't interact with the nucleotide the way that we're supposed to in that active site. Usually that's how it will work. Bind somewhere else on the protein, cause it to change shape, and now our active site is inaccessible. And that's how it will work. But it's non-competitive because we're not at the actual active site when we're doing it. So these are only talking about competitive versus non-competitive, is only asking you about whether or not it's in the active site or not when you're regulating it. All right. These are images, some pictures of what I'm talking about. Okay, so your normal one is this darker color here. It will bind in, yay, enzyme does its work, yay, products. The thing that looks like it, that's competitive, gets bound in there and often can have even a stronger affinity for that active site than the original. And get bound there and get stuck. And now we can't make our product, okay? An example of a protein that can do this, we're coming back to hemoglobin again. At our active site where oxygen will bind near that iron molecule, right? If we bind cyanide there instead, we can't bind oxygen. And cyanide has a stronger affinity for that binding site than oxygen does. So that's why it's so dangerous, why it only takes a little bit. Because it's not getting cleared out and it has to keep up to keep you poisoned. It takes a very little bit and it just stays in that binding site and oxygen cannot force it out. So that's what we're talking about here. Okay, yes. I don't know enough about how arsenic works. I don't know if it like binds with hemoglobin, or I don't know how it works, but it's a good question. A lot of people ask me about arsenic a lot of the time and I don't know enough about it. Look into it, man, let me know, because I'm kind of interested myself. I just never remember to look into it. But anyways, I know about cyanide, and that's why they call it cyanosis, right? If you're turning blue, you're lacking oxygen, has the same outcome essentially as if you were poisoned with cyanide. All right, so we have non-competitive over here. This is where you normally would bind in your active site, and you can usually make your product, whatever. Let's say you build up extra product, that can act as your regulatory molecule, bind somewhere else, and look here, changed the shape, and we cannot bind there anymore, and we won't make any more product, which is usually your regulatory molecule. Although it could be anything else as well. Like in antibiotics, we have antibiotics that work as regulatory molecules as well. All right, repression and stuff like that. We kind of already talked about this. This is just showing you some pictures of how it might work. You get a product that binds on the DNA, and now we can no longer make our RNA stuff like that. So it's a similar concept to what we learned about with our operons, but on any DNA level for anybody, not just bacteria. All right, same thing with induction. It has a similar concept going on there. If we're trying to look at the energy use or making within cells, we are talking about exergonic versus endergonic reactions. Exergonic giving off energy, endergonic needing energy. So think about that exergonic, exo. I always thought of heat. So heat is coming off of your little beaker when I would do the experiment. So it's warm, that's heat being given off, that's energy being given off. If you are having to put energy in or it's sucking energy out of its environment, then that beaker will be cold. So it's just sucking in more heat and that. So a lot of times though, these things will have to be paired somehow to help drive one another. So now we're going to get back into our redox pairs and our redox reactions, oxidation and reduction and everything. They always have to have an electron donor and always have to have an electron acceptor. The things that are going to carry our enzymes within a lot of these processes we're going to get into are NAD and FAD. But first, let's go over redox pairs. If you look at sodium and chlorine, this is how they normally exist. Yes, this is sodium chloride. We're coming back to that. You remember our ionic bonds. Now you can understand why they're ionic bonds. So this is why. Sodium here has this one electron in this orbital outside. Guys, don't remember that outside orbital right there, that third orbital. It wants 8. So 1 is just 1 by itself. It's not happy. Look at chlorine over here, though. Interesting how those pair up, right? So we've got 7 in its outer. It's not happy. It wants 8. But sodium doesn't need that 1. So why does it give it up? To help equal out the balance, right? Chlorine's outer is now happier with the 8. And now we don't have that weird extra outer for any reason for the sodium, because we've lost that electron, but still have the same number of protons. Now we're positive here because we gained an electron, and now we have more than what our protons are. This one's negative. That's why they have the charges that they do. That's why they actually donate their electrons instead of sharing. Okay, so they're happier that way. Keep in mind that if you are gaining electrons, you are reduced. If you are losing electrons, you are oxidized. Don't worry about these agents over here. I'm not asking about those. All right, so a lot of times, always remember, if we're moving electrons around, one has to be a donor and one has to be a recipient. There's never going to be like a loss of it somewhere in there. And this can actually help power our other reactions as we are moving electrons, creating bonds versus breaking bonds, and everything like that, helping create energy in the form of ATP as we're going through this. So let's see. Yes, this goes back to dehydrogenation. The electron transfers in our processes are usually going to be performed in the form of hydrogens being moved around, not H pluses, but whole hydrogens. Okay, so there are electrons in this sense. All right, so NAD versus FAD, and then there's also NADP, which is only in photosynthesis. Yes, we're going to talk about that. All right, these are just our electron carriers. I'm not really going to ask you much more beyond that, okay? All right, so our catabolic pathways are going to be what's going to drive our production, our stealing off, transfer of electrons onto other molecules, okay? So we're breaking bonds and stealing electrons in the meantime. So now we can steal those electrons and we're going to shuttle them down a little pathway, and that's going to help us create energy in a nutshell. But then we'll have to get rid of those electrons. They can't just exist now as free electrons forever and ever and ever at that point. And so for us, when we're talking about aerobic metabolism, aero should always be like air to you, like air that you breathe. That's oxygen. That's going to be your oxygen is your final electron acceptor. For anaerobic, so not aero, these guys use anything else, okay? If it's not oxygen, okay, it's just not oxygen. That's the important part. Okay, so our whole goal is to make ATP. We've already talked about the structure of it, so I'm not going to go back over it, but I am going to reiterate those covalent bonds between those very highly negatively charged, bulky phosphate groups that do not want to be with one another. So there's a lot of potential energy stored between each of these phosphates that if we break those bonds, will release energy. So our goal is to make this which you can imagine has to put a lot of energy into to make it so that the cell can use that later on in other processes. So we have to make a lot of it. Okay, so we can make ATP a few ways. We have substrate level phosphorylation. That just means moving a phosphate from one molecule onto ADP to make it ATP. That's all it means, moving it. The next one is oxidative phosphorylation, which is a series of redox reactions, which is what we're going to talk about. Okay. And then photophosphorylation, go figure, we're going to use the energy of the sun to drive making ATP. Okay, I know we're not doing this right now because I don't have time. Okay, yeah. So we're going to go through 3 different ways that you can make ATP, 3 pathways that you guys have to know the differences of. Aerobic, anaerobic, and fermentation. The biggest 2 that you're going to need to focus on, aerobic and fermentation. We're going to touch on anaerobic, but it's basically the same as aerobic. It's just not oxygen, okay? All right, so all of them use a pathway called glycolysis. And glycolysis is just a process where we break down glucose. Really it's super simple, right? It's really simple, yes, totally simple. You're gonna hate it when I show it to you. Okay, so here is glycolysis in our whole setup. Okay, so we have 3 steps possible. 1, glycolysis. 2, Krebs. 3, this is the electron transport. I call it electron transport chain. The book called it electron transport system. Those are not tricks if they come up as different things. Okay, they're the same thing. All right, so these are possible 3 steps. Now look at aerobic versus anaerobic. What's the same? Glycolysis, yes. What about fermentation? All right, we're good. We're on that same page. Krebs, aerobic versus anaerobic, same, but we don't have Krebs over here. So if you don't have Krebs, you're not going to have ETC either. You have to have that before. They always go together, okay? But see, both of these guys also have ETC, but this one is using oxygen and this one is using non-oxygen. And it gives examples like a sulfate or a nitrate or a carbonate. And you don't even know those. Just know that it's not oxygen, is why I keep saying it, okay? It's literally the only difference. Now I want to point out the amount of energy you're making. Down here, for aerobic respiration, we have up to 38. Cool. For anaerobic, we have up to 36 ATPs. Nice. This is per glucose, per one single glucose molecule for all of these. And per glucose, fermentation yields 2 because it's only going through glycolysis. And your big daddy money energy making goes on here, the electron transport chain. Okay, so we're going over it. So fermentation only uses glycolysis. This is done by facultative anaerobes. It's also done by aerotolerant anaerobes, but I'm not going to ask you about it. Definitely know about the facultative guys because they're going to come up not only in lecture in here, but in lab quite a lot. The whole second part of the semester has quite a lot to do with these fermenting guys. Okay, so if you ferment for all of our intents and purposes, okay, facultative anaerobes. If you ferment, facultative anaerobes. There's no other F's that are going to get confused in there. Facultative anaerobes, okay? Facultative anaerobes. And think about it because your faculty member is telling you it's facultative anaerobes, okay? You could also call them facultative aerobes. All it means is they can switch between. So what happens with these guys is they will go through that high energy producing aerobic respiration on this over here if oxygen is available. If you do not have oxygen, they can't do that anymore, right? But they're not just going to shut down and die like you will. They're going to keep cranking on in the background without the oxygen through fermentation. Okay, so they can switch between. Facultative anaerobes can switch between aero and fermentation. Okay, they make it work. All right, so next is aerobic respiration. We've got to have oxygen here because oxygen is our final electron acceptor. And I'll show you where that happens. But this is what we do, okay? So this is usually what most organisms that rely on oxygen are going to be doing. And if you don't have it and you can still make energy, then you're a fermenter, right? So let's go through these steps. This is glycolysis first. The big important thing, glucose is converted to pyruvic acid. Okay, so glucose, obviously a sugar, we know that. It's a 6-carbon sugar. We're going to go through some steps and y'all get ready, okay? Go through some steps and our end product is pyruvate or pyruvic acid. It just depends on where you are in the body. And this is a 3-carbon. So we're going to make 2 of these. We're just breaking glucose into 2 pyruvates. We're just breaking a 6-carbon molecule into 2 3-carbon molecules. And we're just doing that. We're gonna make a little bit of ATP along the way. It's not important here, but that's only important for the fermenters, right? Because that's where they're getting all their ATP. We'll make a little bit of ATP. But pyruvic acid is an extremely important metabolite, not just for what's going to go on in the Krebs cycle, but for other processes in the cell for building like amino acids and stuff, okay? So glycolysis is extremely important. So this little tiny process where we're going to be going from 6-carbon glucose to our 2 3-carbon pyruvates. This is glycolysis. Anybody happy yet? So up here is your glucose, our starting molecule. And then down here at the bottom is your pyruvic acids. Right here and right here. This is glycolysis. And there's all the steps that it has to go through. Lucky you, I'm not making you learn these. I had to learn every step and exactly where the electrons were going at every point when I took biochem. And this was my first biochem class that I had ever taken, and they made me know that. And I was just like, what are you talking about? Because I have no idea what's happening. I want you guys to get the big picture, okay? It's way more important that you understand that what we're doing is taking the 6-carbon glucose sugar and through, of course, reactions, go figure, moving electrons around. We make a little bit of ATP and look here, guys, look here. What's this? What did we do here? Did we reduce NAD to NADH? We did that too. So I told you those are our electron carriers, right? So we've got a little bit of that too. And those are important as well. We'll get to those in the next one because that's what the importance of the Krebs cycle is.
So it goes through all that just to get to
Yep, yep, that's what it takes. So the reason that it goes the way that it does is because along the way you can make a certain amount of ATP if you do it this way, right? So you lose 2 up here, but you make 2 on each side here. So it's a net of 2 for each glucose molecule. That's where we get it, okay? You don't need to know why it's happening, but I would definitely know, number 1, that it's glucose, I'm just gonna put glue, to pyruvate. That's the big picture. Number 2, you do make some ATP and you do make some electron carriers. In this case, NADH. That's what you need to know about all this BS. We good on that? Hope so. So we've got our little pyruvates. Here he is. Here's our pyruvic acid. He's 3 carbons, 1, 2, 3. He's got some other crap going on around him. And we can use him to build things for the cell, amino acids and all sorts of stuff. Or we can continue on to make more energy in respiration. If you are a fermenter, then you need to do something with your pyruvic acid and those NADHs that you have out here. Those guys, they can't just go around chilling, carrying their electron around. We need to do something with that. And it can't go the normal way, the electron transport chain, because there's no oxygen to accept it at the end. So what they do is they feed it back into pyruvic acid and they make other byproducts by doing that. Things like gas, carbon dioxide, acids, lactic acid, alcohols, and other things can be made too. So here, this is where we're getting ethanol from. You're drinking ethanol. This is where you're getting the bubbles in your beer from. That's your carbon dioxide. This is where you're getting kimchi and kombucha from, and the sourness of your yogurt from. It's lactic acid. It's from that. So that's them dealing with their pyruvate and their extra electrons. Okay. Cool, but we're not doing that because we're aerobes. So we're going on to the next step, which is the Krebs cycle. Okay. The very first thing that we're going to do is take pyruvate and turn it into something called acetyl CoA, coenzyme A, but that's what it is, okay? Pyruvate is a 3-carbon molecule, excuse me, and acetyl CoA is a 2-carbon molecule. This is an important step because of one thing. Where did that carbon go? It's released in the form of carbon dioxide. Anytime you lose a carbon at this step, which is prior to the Krebs cycle, and any step in the Krebs cycle, anytime you lose a carbon, it's coming off as CO2. Okay, so we went from glucose, broke it just into 2 pyruvates. Now each of those pyruvates is getting kicked down to 2 carbons. So now we've got what? We've got 2 carbon dioxides for 1 glucose coming off already. So cool. So what is the big deal, big daddy issue with our Krebs cycle? We're going to be making a lot of our electron carriers. Like a lot of them, okay? And those are important because of the next thing after, all right? What else I want to say about this before we get into that? We do make a little bit of ATP here as well. So what are we making here in the Krebs cycle? We have taken, this is the Krebs cycle, y'all. Pyruvate, which is 3 carbon, down to acetyl-CoA. This is the only part that I need to be familiar with as far as what's happening, because we're going to give off carbon dioxide here. Okay, then acetyl-CoA goes into Krebs. And then ultimately, it's just going to, everything's getting cycled through. We have the stupid 2 carbon combining up with this already existing 4 carbon guy. Okay, so now we're back to friggin' 6 carbons here. This is 6, this is 2, this is 4. I'm not gonna make you know the steps. I just want to point out what's happening here. Okay, so we've got 6 carbons here. And then each step that we go through, we are moving around electrons and possibly losing carbon dioxide in the process. Just depends, okay? So as we're doing that, we're making hella NADH and kicking off carbon dioxides, okay? Here's another one, carbon dioxide. And you can count it too, because this went from 1, 2, 3, 4, 5, 6 carbons. Now this is 1, 2, 3, 4, 5. That's where it went. Then this is 1, 2, 3, 4. That's where it went, etc. Right? And we can keep track of that. Along the way, while we're moving all of our things around and stealing off as many of these electrons as we can, we make FADH2, NADHs, and a little bit of ATP down here, a little bit. So we're going through this whole thing. That's the Krebs. What are we giving off? We're giving off a little bit of ATP. We're giving off our electron carriers. Remember, that's going to be NADH and FADH2. I don't need you to know the numbers of them either. I don't care about that, okay? And then also our byproduct of carbon dioxide, okay? That's what you're getting from the Krebs cycle. Just by moving those carbons around in that cycle, we can get all this stuff. So the big deal here, okay, we made a little bit of ATP. Who cares? We made a little bit of ATP in the glycolysis. Who cares? The whole point of glycolysis was to give us pyruvate, right? That's the big deal of glycolysis, because now we can shuttle it through the Krebs cycle. The whole big deal of the Krebs cycle is to make our electron carriers. That's the big deal of the Krebs cycle. So now we can shove it through the electron transport chain. It's our 3rd step, right? I told you it was coming. It's going to be glycolysis, Krebs, electron transport chain. This is our electron transport chain. We're going to shove NADH's electrons and FADH2's electrons through, they say, a series of redox reactions that you don't need to know, but that's what's happening, right? We're moving electrons. If you're moving electrons, that's a redox reaction. And while we're doing that, it allows us to pump hydrogens across the membrane and all this stuff. So let's go through it. So your little tiny NADH guy donates its little electron into these guys, okay, these proteins. And by introducing that negative electron, essentially, and changing the shape and exciting these guys with that electron, now we're going to pump our leftover H+, because we're literally putting electrons through. Okay, not talking about hydrogen-based electrons. We're talking about taking the electron and shoving it through, okay? So the electron's going through, yay, electron is moving through here, electron, all the way through these guys, right? As we're doing that with these negative electrons, these leftover positive hydrogens are getting pushed across to the other side of the membrane. Now, in case this doesn't look familiar to you guys, this is our gram-positive peptidoglycan cell wall. And this is our cytoplasmic membrane. All right, so that's just the membrane that touches the insides of the cell. So that's where this happens in bacteria. So where do you think it happens in your cells? It happens on the folds of your mitochondria, your what would be the cytoplasmic membrane of a bacteria. So the inner membrane of that, which is all those folds, those cristae in your mitochondria. Okay, so that's where it happens in your cells. This is where it happens in bacteria. So anyway, so we've got these okay, we're getting there, I promise. This is really important stuff, so I want to be sure we get through it. This is where we get the negative guys moving across, and then we have the positive hydrogens getting moved out. So now we're creating a gradient. So we have this buildup of this gradient on the outside of that membrane, right? Like a dam. It is like just a ton of pluses on the outside, right? And there's a dam in place. And there's one place for them to come back through so they can try to equal out the dam level. And that is through this portal, so to speak, this protein called ATP synthase. This molecule here, it's a channel. And as these positively charged guys move through that protein, because it's just a protein, and we know how positives can affect those, right? As they move through there, this changes its shape and allows for ADP to be phosphorylated into ATP. So we have to have those protons, those H pluses, moving through and moving through and moving through constantly. But that's a problem, right? If you have too many of them coming through onto the other side of the dam, what happens if it evens out? That's no more ATP, right? So we have to do something there. We have to do something with those H pluses as they come in and the electrons, because we can't have them just hanging out either, okay? So those electrons that have moved through the system, let's say we'd have 2 electrons, and we'll take 2 H pluses, and we will take half of an oxygen. And what do you think that makes? It's not a mystery, guys. It's H2O. Look at that. Okay, that's what we're doing with it. We've consolidated everything. So oxygen comes in as a way of getting rid of the H pluses to maintain that gradient, of getting rid of the electrons that have moved through those proteins. They're called cytochromes, that have allowed us to create the gradient. And now we can keep cranking because we can maintain that, all right? So letting things through the dam, but not holding another dam on the other side, letting it flow down the river essentially, right? So we're not equaling out on both sides. That's what's happening here. So we can create the energy for our cells. This is a system called, like it says at the top, oxidative phosphorylation, because we're allowing phosphorylation to happen because of this electron exchange. Okay, so if we look at this, our oxygen that we are using up here, this is the only place in your entire body where you breathe in oxygen and it goes. The only place in your entire body. You breathe in right now, it's all going to that. All of it. All of the carbon dioxide that you breathe out, right here. All of it. You breathe in and out because of that. That's the only reason. There is no other reason. You go for a run, you breathe harder. Why? Because you need more energy. You need more oxygen to take care of that H pluses, right? The carbon dioxide that you breathe out, the CO2 that you're breathing out, the oxygens that are part of that CO2 are not part of the oxygen that you breathe in, right? They're part of your food. You're breathing out your food, y'all. So this is why I like to get to this part, and I know that we're going late, but if I don't go over this, this is one of the most important things you can possibly learn about how the body functions, I feel like, as far as like where your energy comes from, why you even frickin' breathe. This is why you breathe. So it's pretty cool stuff. I'm aware of that. Okay, anyways, so oxidative phosphorylation and the means by which that is happening, I am going to be done, I promise, is called chemiosmosis. So moving those H pluses across because of those electrons, that's chemiosmosis. That will be on the exam. That's why I'm saying it. And when we have that buildup of the H pluses, those are called the proton motive force. That's giving the motivation for us to make ATP. The protons are. Okay. We already talked about this. We already talked about this. I'm gonna share. This is a good summary slide if you want to come back and see how much of what is made in what step. Okay. Most of our ATP is made in that last step where we have the electron transport chain. Okay. 34 of it is made there. So cool. All right. So we depend on oxygen. Am I missing anything besides? We already talked about this fermentation, how we're getting rid of pyruvic acid. Fats give more energy than carbon. That's why your body stores energy as fat. So yeah. So you get 50 ATP from a fatty acid that's 6 carbons, whereas you only get the 38 from a carbohydrate like glucose that's 6 carbons. So it's pretty crazy, but it's just an amount of energy that's held in the bonds there, but it's more efficient for your body to store it that way. But that also means that, you know, there's a lot of energy sitting there that you might have a hard time burning through. So whatever. You can also break down proteins, enter stuff in. Metabolism is talking about if you're making intermediates, stealing stuff from the in-between parts to use it to build other things, or using parts from building other things to shove it into making energy. That's all. And we'll talk about photosynthesis another time. I don't want to go any farther. It's only a couple more slides, so it's not like a big deal. I didn't realize how long it was going to take this morning, so